Radek Nowak is a professor of immune engineering and drug discovery at the University of Bonn and the steering committee member of the Immunosensation 2 Cluster of Excellence in Bonn. Prior to joining University of Bonn, Dr. Nowak completed a postdoctoral fellowship and became a senior scientist at the laboratory of Professor Eric Fischer here at Dana-Farber. And a group leader and a member of the leadership team at the Center of Protein Degradation at the Dana-Farber Cancer Institute in Boston. Dr. Nowak research focused on development of molecular glues and characterization of novel mechanisms of degradation. Welcome back to this webinar series, and we are very much looking forward to your seminar, Radek.
Thank you so much, Mikołaj, and yeah, thanks so much for this very kind introduction. And I'm very happy to be here, although on the other side, and also on the other side of the pond here in Bonn. So today I will be talking about structure-based design of covalent cereblon binders, glues, and PROTACs. But I think no targeted protein degradation talk would be unnoticed without introduction. And I think for those of you that are perhaps new to the field, the ubiquitin proteasome system is a very interesting cell biology system. So it starts off, it's effectively a way of targeting proteins with ubiquitin that results in different cellular functions. So it starts off with an E1 activating enzyme that turns over ATP to ADP and charges ubiquitin, transfers that to an E2 conjugating enzyme, and that E2 subsequently binds an E3 ligase, and then ubiquitin is then transferred to a substrate. And that transfer of ubiquitin can be on multiple different fronts. So you can effectively create a polyubiquitin chains, and this is effectively the signal that results in tagging protein for degradation by proteasome. You can also have multi and monoubiquitination that results in many different signaling pathways. So now if you look at how these ligases are built and coming from a structural biology field, this is effectively how a Callind ring ligase is made out of. So Callind ring ligases, there's about 200 of them in the E3 ligase family. And it starts off with a Cal4 or Calin arm, to which you have an adapter protein, in this case DDB1, that consists of beta propellers, in this case 3 beta propellers, one of them that binds Cal4A. And you also have a substrate receptor that happens to bind, in this case, a drug, and that drug is called thalidomide, and I think many of you will be familiar with that compound. So thalidomide will be recruiting transcription factors for degradation, and then substrate will be placing itself just next to thalidomide itself. The E2 would be binding close to RBX1 and then discharging ubiquitin to the substrate by just sheer proximity. So if we now look at some of these small molecules like analogs of cereblon binders like thalidomide, EM12, lenalidomide, and pomalidomide, they vary only by a couple of different atoms, but what they are able to do is they are able to effectively recruit proteins for degradation through binding of this beta hairpin loop noted here in casein kinase 1 alpha structure. And what is quite special about cereblon is really the amino acid composition of the binding site. So the binding site is composed of tryptophan pocket, but outside of that pocket, there's a couple of really interesting residues. And I wanted to draw your attention to this histidine 353. This residue we have shown happens to be reactive, and we are able to effectively design covalent compounds targeting that residue. So now it brings me to a very interesting set of chemistry, sulfonyl fluoride exchange chemistry, which happens to be quite useful as a chemical biology platform. So this is effectively a set of a chemical warhead that effectively allows you to target different amino acids covalently, and that goes for tyrosines, lysines, serines, threonines, and also histidines. And the way this works is through nucleophilic substitution. So the leaving group would be hydrogen fluoride gas, and that's effectively how you can label many of these different amino acids. So sulfonyl fluoride exchange chemistry is a great way to effectively label proteins inside cells through chemical probes. One thing that these compounds are really good at is they tend to have really good water solubility and water stability, and really don't perturb the function of the molecule, don't perturb the lipophilicity of the molecule. So one thing that we wanted to effectively ask, and this part of this work has been presented by Ellen Jones here in the audience not so long ago, we were basically asking our question: can we rationally target the histidine residue? And just to take a step back, there are examples of compounds like FSBA, for example, which has been used to study kinases, that happens to effectively covalently label a histidine in the active site of one of these enzymes. And this is effectively serendipitously discovered, and there has been no way to effectively target histidine in a rational way. So to do that, and this is published work in RC Chemical Biology, we effectively started off designing a set of IMID molecules that happen to have these covalent warheads on. And here I wanted to draw your attention to two sets of scaffolds. One of them is a sulfonylfluoride molecule, and the other one is fluorosulfate. So fluorosulfates happen to be significantly less reactive than sulfonylfluorides, but if you look at a nanobright engagement assay across the whole family of these compounds, they happen to be very potent engagers of cereblon. And in this particular case, sulfonylfluoride molecule happens to be sub-nanomolar engager of cereblon. So subsequently, we are able to show that this fluorosulfate molecule, and this is the less reactive molecule, is actually able to label cereblon covalently. And this is recombinant cereblon that we are able to covalently label with the molecule upon a treatment. And we are able to identify the peptide that happens to be labeled. And in this case, this corresponds to the histidine 353. Interestingly, the labeling is selective. So if you look at BSA, you're not able to label BSA despite the presence of multiple surface exposed tyrosines or serines or residues that would otherwise be reactive to sulfonylfluoride chemistry. So this compound, when we tested it in proteomic studies, actually happens to be rather interesting. It happens to be quite a nice inhibitor of cereblon and doesn't happen to degrade any proteins. So we are effectively able to use it as a covalent cereblon inhibitor and rescue degradation of IKSDIV1 by lenalidomide. So if we now look at additional derivatives that happen to effectively engage this histidine 353, they also happen to label cereblon in a similar manner. We are also able to see histidine 353 labeled in this case through proteomic studies. And then now if we look at, in this case, the fluorosulfate derivative, we are able to see that one protein happens to be degraded, and this is NTAC1. So NTAC1 happens to be the only protein that happens to be significantly degraded in this proteomic study, and that was done in two different cell lines, MOLD4s and THP1s. We are able to subsequently confirm that in a Western blot, showing that we can actually degrade NTAC1, and the degradation of NTAC1 happens to also be methylation dependent, so there's proteasomal action here. NTAC1 is one of these components of an end-root pathway, also involved in protein degradation. So overall, what we are able to do in this past work is create two types of compounds: covalent cereblon inhibitors, and those are the sulfonylfluoride molecules, and fluorosulfates, which happen to effectively glue cereblon to, in this case, NTAC1. And both of these molecules label histidine 353. In subsequent work that has been recently published, we are also able to show that we can actually substitute the fluoride in this case for an imidazole, and that happens to be actually a very good leaving group, which also allows us to maintain cereblon engagement and also really nice plasma stability, human liver microsome stability, and human hepatocyte stability comparable to the parental molecule EM12. But what I wanted to talk to you today about is design of covalent cereblon PROTACs. So we were basically wondering if we can take this idea of covalently engaging histidine 353 and then develop molecules that would be able to target that histidine 353 and therefore maintain occupancy on the ligase while being able to recruit novel targets through PROTAC moiety. So we focused on this molecule ARV-825, and this is a known BRD4 degrader presented by Arvinas, and we put fluorosulfate on the solvent exposed position, hoping that that would engage histidine 353. So that fluorosulfate PROTACs happens to label cereblon, and actually you can see that labeling of cereblon is more or less 50% complete after 24-hour incubation with 10 micromolar of this molecule. And with the help of Scott Ficarro and Jared Marto at the Dana-Farber, we are also able to actually map the site of this labeling that happened to be the histidine 353 of cereblon. Interestingly, the way we have identified this peptide is because peptide, when it was fragmented in the mass spec, happened to result in production of this JQ1 ions that allowed us to identify nearly all of the peptides that happen to have this labeling in there. So that forms a very interesting biomarker for identification of these covalently labeled peptides through this particular chemistry, and especially when we think about JQ1 there. So now we wanted to assess its activity in cells. And the way we've done it is we compared it to two available PROTACs, DBET6 and ARV-825. So the one thing we have done is we have done a competition experiment with a very potent cereblon engager, and that is CC92480. So when incubating with CC92480, we're able to effectively quench degradation by DBET6, and this is in a hybrid BRD4 cell line and HEC293 T cells. Same with ARV-825, although perhaps to a lesser extent. But the activity of fluorosulfate ARV-825 was not affected at all, indicating that covalent reaction has taken place, and we are actually looking at, we are unable to compete it because of covalently labeling cereblon in this case. So we further probed this modality of covalent engagement of the ligase with a washout experiment. And this is a rather simple experiment. We have taken hybrid BRD4 cells, treated them for 5 hours with a PROTAC molecule, and BRD4 would be degraded at this point, then 3 times PBS wash, and allowed for a 24-hour recovery. In the premise, we replaced the media with addition of CC92480. So that allowed us to fully block the ligase again and looked at what happens to be the resynthesized form of BRD4. So if you perform this experiment with DBET6, you see exact same thing. The PROTAC happens to be quite nicely rescued. So we observe no degradation, almost no degradation upon a washout. Similar with ARV-825, although there is something happening, and we definitely see much longer or like maintained DC50 value for this fluorosulfate ARV-825 molecule, but perhaps slightly reduced depth of degradation. So the other thing we wanted to look at is we wanted to look at kinetics of degradation. And in this case, we also compared these 3 molecules, DBET6, ARV-825, and then the fluorosulfate PROTACs. And we were able to show that kinetics of degradation happens to be actually more or less the same for ARV-825, between 5 and 24 hours, perhaps a little bit more of a hook effect at higher concentrations. And for the fluorosulfate ARV-825, we have seen that the depth of degradation is affected, but the DC50 is maintained, indicating that perhaps compound requires a little bit more time to label cereblon and become active for degradation of BRD4. The other explanation, or the other way of actually looking at it, the activity of these molecules is through cerebral engagement. So the one thing that is actually quite interesting for this fluorosulfate molecule it happens to have a much weaker cerebral engagement than this protoplast molecule ALV-825. And that molecule happens to be about 100 nanomolar in potency, whereas our PROTAC molecule happens to be around 1 micromolar. We were able to quite nicely follow that up through Kinetics study with a live cell substrate, and we were able to follow the IC50s with the nanobread cereblon engagement assay. And you can see that it happens to be effectively rather slow for fluorosulfate ARV825, but more or less around 20 minutes, we are able to see pretty much equilibration of the assay. Whereas for the parental molecule, ARV825, it's pretty rapid kinetics, rapid engagement of cereblon, and really maintained over a longer time. So finally, it would be excellent to know what selectivity of this molecule happens to result in proteomic study, and this is something that we have tried, and we have performed a longer experiment, in this case a 24-hour experiment with 1 micromolar of the fluorosulfate ARV825 in mold-4 cells, and we are able to see degradation of BRD2, 3, and 4, and then probably because of this long experiment time, we are also able to see MYC going down and P53 going up. So these would be effectively consistent with degradation of BRD4 or BET family of proteins. We also looked at off-target selectivity of this molecule, and in this case, we compared it to known GSPT1 degraders in our ultrasensitive GSPT1 GFP fusion assay. So we see now CC885 to be really a picomolar degrader here, and CC9009 about 1 nanomolar degrader. We see some degradation of GSPT1 with the fluorosulfate PROTAC, but these concentrations typically don't correspond to very strong degradation in the native cell line. We also looked at hybrid ICSDIV1 assay, and we see that while ARV825 is able to actually degrade ICSDIV1, it's substantially reduced in the fluorosulfate ARV825 setting. So on that note, what we are able to effectively show is a whole spectrum of covalent cereblon modulators. So we have the covalent cereblon inhibitor, and I realize I think I have mixed up the slides in the very beginning, so please excuse me for this. It's the fluorosulfate molecule that happens to be covalent cereblon inhibitor, and then sulfonyl fluoride, pardon me, and then the fluorosulfate happens to be the NTAC1 molecular glue degrader. But now we were able to develop a fluorosulfate version of a PROTAC molecule, enabling effectively cereblon for discovery of covalent PROTACs. So that could result in prolonged degradation, prolonged engagement of the ligase, as we have shown in this study. So this kind of brings me to this short description that what we are able to do effectively here is chemically modify the ligase, and in this case we are chemically modifying cereblon to effectively cause degradation of novel neosubstrates NTAC1. We have seen that through proteomics, we have seen that through cereblon intact mass, which is very similar to what we can actually do with this genetic modification, and that work has already been presented within the TPD webinar series. And it's the mutations on the ligase KBTBD4 that also happen to induce a neomorphic function of the ligase, degrading co-rest complex. So we are able to now effectively do these through covalent modulation, through reversible modulation, but also genetically. So I think it nicely completes the picture. And I wanted to also outline these molecules, which I think are really quite exciting, and in my mind, they do very similar things. They effectively are able to recruit novel proteins for induced proximity, and they happen to have very interesting molecular mechanism of action. So on that note, I wanted to take a minute and introduce you to what I want to do in my new lab. So as Mikołaj have mentioned very kindly in the very beginning of the introduction, I have moved from Boston to Bonn across the pond to Germany. So I'm part of University of Bonn in the Institute of Structural Biology. And you can see this is effectively the morning and the afternoon shot of the institute. It looks really quite nice. This is where the lab is located. So if you visit, you're going to find us there. And just to briefly outline what I'm thinking in terms of what we want to do in the lab, we are looking at interactome research and protein engineering using small molecules and synthetic biology. So the overall idea is that, as I mentioned before, proteins would bind small molecules or covalently be labeled by them. They would form a neoprotein, which would have a neoprotein interactome. Some of this interactome would be functional, and the function could be degradation, but it could also be way beyond degradation, as we have observed through recent publications. It could range in changing PTM modifications, chaperone deprivation, many different functions. So this is something that I wanted to explore extensively. And the techniques we will be using within the lab range from synthetic biology, strong focus on immunology, and this is an excellence really in Bonn to be able to do that and be embedded in this environment, cancer biology, structural biology through cryo-EM and crystallography, and trying to really design and synthesize some of these molecules through collaborations and also within the lab through screening and through chemistry. So the group at the moment is growing. So we have the first PhD student started, Jan. Hello, Jan. Jan has started in December, and we have another person starting, Dominika, who's starting in February as an MSc student. And we are actively recruiting. So if you are interested in a postdoctoral position or a technician position or a PhD position later on, please reach out. And I will be happy to talk to you, and especially if you're interested in designing molecular glues. So on that note, I wanted to really thank you for the attention. I wanted to thank Lynn Johns, Ben Ebert, and Eric Fisher, Nathaniel Gray from the Center for Protein Degradation, and majority of that work has already been presented, and novel work has been published. So that has really, it's been a tremendous pleasure to work with people there, and I really like the environment, the atmosphere. So you guys are great. And on that front, I also wanted to thank my previous lab, Eric Fisher, and all of the people within the lab who were able to help me out along the way, and especially through this transition to a new position. So I'll be happy to answer any questions. And